That was such a kind introduction, and I think I even wrote it myself. It's so nice. Oh, look, the slides work. I don't have to do an interpretive dance now. I'm delighted to be here today. I spent so many years of my life on this campus, and it's always a delight to be able to come back. The last time I was here, we were talking about raising money for what is now a gaping hole just on the other side of University N, which I was thrilled to see. And I'm so looking forward to coming back and seeing the beautiful building that's going to stand on that spot. So when I was thinking about what I wanted to talk about today, after I received Travis's very kind invitation, we discussed it, and what we talked about was maybe a type of a career discussion. But it's not going to be, I think, your typical career discussion, because I think my career has not been incredibly typical. And what you'll see as I go through my different slides, just so I have something to point at and gesticulate wildly, is actually very similar to the types of discussions that I have with students. And so maybe we can consider this a broad advising discussion. And before I get going, I have to tell you one thing, since this is an advising discussion. I tell my students, if you make eye contact with me, it means you want to tell me something. Now, this can lead to whiplash in a certain number of cases, as people dodge and try to look away, and they look everywhere but at me. But this will be really boring if it's just me standing up here and we don't have some things to discuss. And I'm actually going to ask you a couple of questions as we go through. We'll see how many slides we get through. If we don't get through all of them, well, then so much the better. So what I've entitled my presentation as is Things I've Learned Since I Left the Farm. This is the farm I grew up on. That's my mother, who now lives in a memory care facility in Taylorsville. She has non-Alzheimer's dementia. She doesn't even remember living on a farm. She doesn't remember any of her kids, but she's still a very interesting person, loves old redneck music, which I sit and play to her. I'll do that Friday after I'm finished here on the campus. But I'll just say at the beginning, it's a very lucky kid that grows up on a farm. It's very important, I think, to state my conflicts of interest in advance. Unfortunately, I don't have any. I hear they're very lucrative. I'm always on the lookout for some, so if anybody has any ideas, just give me a shout out. And it's important to state, because I'm an employee of Harvard University, that the views expressed here are my own. I'm not here on behalf of the university. I'm here because Travis invited me. And thank you for the invitation. Okay, you can't tell the players without a program. We're not going to talk about very many things in terms of the variety. This is the most interesting part, the wild and reckless speculation, so I hope we get to that at some point. I think it's important to know where the floor is. What is your foundation? What is it that you stand upon that gives you strength so you can rise and greet the day, or the different challenges that you have in your life? This is my foundation. These are my people. This is my son Dante. He's named for his grandfather Dante, who's named for his father Dante, all the way back to Firenze. And my daughter Annie. And this is my favorite librarian, Andrea Fiorillo. Her name means little flower in Italian, and it's true. So, knowing where you're from, to whom you're connected, I think is a foundational part of life. Gesundheit. There are other foundations in this picture that you can't see, so let me illustrate a few. We're a very academic family. We love to read. We love to study. My boy is at Cambridge Rindge in Latin. He just started 10th grade this week. This is what he looks like probably today, having started school this week. My daughter Annie attended Pacific Northwest College of Art. She's now a production assistant for Fire and Ice Productions in Park City. They're working on a secret project. She won't tell me anything about it, except that Kevin Costner is involved, and it's something about a ranch outside of Yellowstone. So I look forward to seeing her name scroll by at some point in the future. My wife studied religion and dance at Lewis and Clark College, and then went on to Simmons College, a terrific small college in Boston, probably one of the country's foremost programs in library science, and she is my favorite librarian. She's an adult services librarian at the Reading Public Library, where she combines her incredible call to public service with outreach and helping people at an individual level. I'm a bit of an academic overachiever in the family. We're going to talk a little bit about some of these as we go through. And I'm not just saying this because of where I'm at. The most important institution on this chart is that one at the very top, Utah State University. I would be nowhere if not for this university being in my life, and I'm very grateful. Now, I want to tell you one thing. Well, I'm going to tell you several things. And as we get to know each other, you'll see that I'm full of stories. This lady is a very important story. I want to just tell you briefly how we met. I was actually going to a party with a friend of mine when I was in grad school, and she agreed to take me to the party as long as I wasn't a pain, which was a difficult thing for me to promise, but I promised nevertheless. She had to stop at her house before we went to the party, and so I said, I won't even come in. I'll wait in the car. I won't touch anything. And as I was sitting in the car, I saw this amazing person walk by the window. And it was Andrea. And I went inside and introduced myself. And my friend went to the party without me. And we've been together ever since. And I think that this is a lesson that I learned at an early age. I'm going to tell you a few things that I know, and you'll see how my personal life interdigitates with my professional life. But this is an important one. It is a whole lot easier to live with failure than regret. If you see an opportunity, if there's something you want to do, don't be the reason that you don't do it. Give it a try. You may crash and burn, but it's so much better to have crashed and burned than to say, well, I wonder what might have happened. This is a principle that applies through many things that we're going to talk about. Who are my people more broadly? These are my grandparents, Wilhelm and Anna Lensch. My grandmother was a mail-order bride from Berlin. This is the wedding photograph. It was also the day that they met in person in south central Colorado, after my grandfather, who came to this country with his brother Matthias from Germany at the early part of the last century. They looked pretty happy there. They had 3 kids, including my father, Martin Lensch. He was a cowpuncher. He grew up ranching in south central Colorado. He essentially ran away from home, from what I remember, when he was about 13 and bummed around on ranches until the world changed around him, and he changed with it. My father, from what I remember, he died when I was 15, but I remember him telling me that he got as far as the 5th grade, which was considered a great deal of education where he was from, and at that time he was born in 1917. But because of the war, he obtained his GED, and he became an airplane mechanic, which I think was pretty amazing for someone with a 5th grade education. Now, he never kept up as an airplane mechanic after the war, and one of my great regrets is that I never thought to ask him why when he was still around, but I think it had something to do with the fact that his formal education was as far as the 5th grade. My mother, Dolores, whom I mentioned briefly, is still alive. She's a good old farmer lady. I come from pretty rough stock. I'll just leave it at that. I grew up in Lehi, Utah. It's a familiar sight, right? Lehi's major industry, at least it was when I was growing up. Some quick things. I'm the youngest of 9. I had wonderful and dedicated teachers in my hometown of Lehi. I imagine you had the same. I got mostly good grades, but not always. Especially after, right around the time that my father died, life was pretty rough at home. And maybe suffice to say that I didn't exactly come from a house where academics were highly valued. I've advised many students over the years, and the students that I choose to advise at Harvard are all first-generation students because we have a lot in common. And I've met students whose parents are delighted that their kids go to college. I've met those who are horrified that their children are going to college. And then I meet those in the middle, and mine were more or less in the middle. They weren't necessarily in my way, but it wasn't something that was important to them or that they valued. I love to read. Couldn't get enough of books. Books are a time machine. They're a teleporter. They're just an amazing thing. It's probably why I'm married to a librarian, come to think of it. I never made that connection before. I also love science, exploring, and music of all kinds. I remember one of my favorite memories was I had this Ray Charles record on at home, and my dad said, "Why can't you just listen to normal music?" Well, I thought that was pretty normal music. It was exciting. It was very exciting to me. Maybe that little bit of foreshadowing into, as I looked around at where I was from and the people I knew, they were good people, hardworking people, but they weren't doing the types of things that I felt I wanted to do with my life. I wanted to study. I wanted to go out into the world. I didn't know what I wanted to do when I got there, but having that perspective of saying where I'm from is good, but it's not quite me, I think is important. And that it kind of comes back to that point I made earlier about it being easier to live with failure than regret. Not everyone in my family had graduated high school. Neither of my brothers above me did. I'm actually not sure if my mother graduated high school or not, though all of my sisters did. And nobody had gone to college. College never even came up at home. It was just not part of our conversation. So I aspired to go to college, but I had no way to get there. I just had no idea. I liked to take tests. I really was interested in applying myself, but it's awfully hard to figure out what to do if there's nobody to help you get there. Until this time last year, I met this man. I had never met him before. I didn't know he was going to be there. He didn't know I was going to be there. This was at the Aggie football game. I won't say who we played because it was an embarrassing game. I think we lost by like 300 points. There were like amputations during the game. It was awful. As he was leaving, I figured out that his parents owned the newspaper in my hometown. Sorry, it's just amazing to think that I ran into him, because he's my hero. His parents gave me a scholarship. They'd never met me. I don't even know how they found out about me. I got to go to college because of his parents. Now, it wasn't a full freight scholarship. It wasn't a full ride. It was actually pretty small. Utah State was a lot cheaper in those days, but it was a $400 cash scholarship. It was enough to start. I mean, you can sit in a canoe on the bank all you want and just paddle as hard as you can, but you're not going to go anywhere. You need your start. You need that push. This is Wes Ennis, and his family gave me that push. They gave me the gift of my own future, and it allowed me to come to this great campus and to study, and I'm very grateful. This I know. Ability is insufficient for success. It must also be wedded to opportunity. The Ennis family gave me an opportunity. Others in this room gave me opportunity as well. I plan to talk about that a little bit. But here I came, an Aggie, and I should say I am a true Aggie. I was made a true Aggie by Miss Bluffdale, which is where the prison is, so that's saying something. I came to college. I majored in biology. Emphasis in microbial genetics. I think I took every genetics course that was available here, including from the late, great Jim Bowman, one of my favorite professors ever. College? College was hard for me. I didn't know anything about college. I mean, to be quite honest, it wasn't until years and years later, until I began to advise students, that I think I actually figured out what college was supposed to be. I certainly didn't understand it during the time I was here. So it was tough. I actually withdrew twice, and somehow each time I made it back. So I have one of those 6-year degrees, although I don't have a master's, I have a bachelor's degree. But in 1991, I accomplished something that no one in the history of my family had ever done. I got a college degree. I was the first, but I think when it's your turn to be first, it's very important to look for who is second. And I'm delighted to recognize my niece, Willy Lensch, who's also a proud Aggie, who's here today. I'm so proud of you. Thank you for coming. She's going to graduate soon and is going to be a fabulous nurse. Sorry to embarrass you, it had to be done. I was fortunate to study under some terrific people, some of whom are here today. One of them was this fellow, the Slime Lord, Dennis Welker. I studied extrachromosomal DNA in his lab as part of undergraduate research projects one summer, along with Dr. Hughes, his partner in science as well as life. He taught me some incredible things in your laboratory. I think foremost among them was how being focused on a specific topic and dedicating yourself to it can actually be an incredibly revealing experience, a joyful experience, and that with the hard work that comes from being dedicated to a task, amazing other things will arise. So thank you for teaching me that, both of you. Pretty common sentiment. Okay, I'm getting back to work. There are others. This is one of my favorite professors ever, who's also here. Gratias tibi ago, eruditus. This professor, Mark Damon, who was my Latin professor. What did Mark teach me other than Latin? As best as he could. He taught me that nothing under the sun is new, which is the truth. But importantly, Mark taught me that to explore is to learn. So I learned about focus in Dennis's lab, but I learned studying as your student that understanding a broad array of topics is actually enriching to the person, and it stimulates your thinking in lots of ways that you wouldn't anticipate in advance. So thank you for teaching me that lesson. Thank you for coming today. So after college, I did something that's actually pretty typical. I decided to take a gap year. Well, my gap year became a little bit more than a gap year. Great advice from professors here, including Greg Podgorski, who's here today. I was his student in developmental biology. Learned some really great stuff about somites and so forth under Greg's tutelage. I applied for a position at the University of Utah and got one working in a very small lab. There were 3 of us, including this gentleman, Phil Chance. And this was during the time that the Human Genome Project was really barreling away. And we were working very hard to identify genes that caused human neuromuscular diseases. And it was kind of a theme was developing over the years. And this was actually very successful work. By the time I finished working with Phil, I had been a co-author on 8 papers and had a patent that I still continue to receive a little bit of royalty money on. It was a very rich environment, and I was happy to work there. But since we're lecturing, I have to give you an example of what I mean. So many things in the lab that turn out to be pretty fun start with saying something like this. I wonder why that happened. And most of the time it's because you made a mistake, but once in a while it's because you're on the precipice of learning something about the world that nobody else in the world knows. There are few feelings in life as satisfying as that moment. And I want to give you an example. So we're going to look at a pedigree here. So for those of you not anointed in pedigree analysis, guys are squares, women are round. I didn't make that up. That's how I was educated. And if it's shaded in, that means the person has the disease that we're studying. This particular disease would be called hereditary neuropathy with liability to pressure palsy. It's also called Dutch bulb digger's disease. It has a lot of different names. And it essentially, the people who experience this have a rare type of demyelinating peripheral neuropathy. Myelin is like the insulation that goes around your nerves, and sometimes it would go away in these people. And where they would be particularly vulnerable is places where your nerves are exposed, like your funny bone, or here, you know, where it gets smacked to see if you have a reflex. If a person with this disease hits their funny bone, that feeling when your arm goes numb for 15 or 30 seconds could last for days because they don't have myelin around areas of that nerve. The nerve is exposed and it experiences damage. So we were very interested in understanding the genetic basis of this disease. So the family comes into University of Utah, we draw blood, I isolate DNA, and we start to study them. And then something interesting happens. Now, this is just an arbitrary way of labeling alleles. And what we're doing is looking at the identity of the chromosome that the mother gave and that the father gave. Now, there's something peculiar about this. Look at this for a second. And again, like in this woman, maybe this A came from her mother and the B came from her father. So genetically, she resembles both of them. Same with this child. The father up here. These folks are heterozygous. This one's homozygous. AA. What's the problem here? Oh, yeah. And be a little bit more specific.
Usually in situations like this, the dude gets, like, I can't think of the term right now, but it would be from the father and the mother.
And what don't you see here?
An A.
Oh, this is scandalous. Look at this. In genetics, this is called a misinheritance. And when you do a lot of genetic studies and you get a large pedigree, the ones with 3 different generations and maybe 20, 30 or more people, you'd be surprised, but about 1 in 5 large families like that have a misinheritance. And it usually means that somebody's been away on too many business trips, if you get my drift. So that's what you would assume in this case, except let me make it a little more interesting. Oh, the person where there's a misinheritance has the same rare disease. So it's pretty unlikely that that little girl is not related to that gentleman. So what could explain this? Think about that for a second. Yes, sir, what do you think?
It's on a different gene.
Sorry?
It's on a different gene.
It's on a different gene. Well, at this point, I actually don't even know if I'm studying the gene that causes the disease. All I can tell is that by comparing their DNA, they don't appear related when I look at the alleles. But when I look that they both have this rare disease and they resemble one another, they do look related. So that's a paradox. Think about it for one more second. What do you think?
Like a really specific mutation?
A very specific mutation. What kind of a mutation, maybe?
Epigenetics?
Well, it could be epigenetic, except for the fact that the DNA itself still doesn't match. It's a bit of a poser, isn't it? What do you think?
Something like
Gene
gene conversion?
conversion. Interesting. So that there's been a duplication of one strand onto the other strand. I'll tell you what my boss thought. He thought I switched the samples. And he said I was an incompetent lab technician. And I said, no, I work really hard. I'm not a moron. Whoever drew the blood's a moron. And he said, I drew the blood. So I'm like, oh, this is not going to turn out well for me. Let me tell you what the answer is. The answer is this. They're not actually homozygous. This person is not an AA, they're an A nothing. This person is not a BB, they're a B nothing. There's a large deletion on one of their chromosomes, and I actually wasn't detecting it at all. I thought I was looking at both chromosomes, but I was only looking at one. The very first experiment that I did actually revealed the very genetic basis for this disease. And it ended up being a 1.5 megabase deletion on this chromosome. We were able to go on and show that it was exactly the same genomic interval that when it's duplicated causes another human disease, and it was the first example ever in the history of science of reciprocal duplication and deletion syndromes in human genetics. I almost got fired for it. But I believed in the way that I was doing science because I was trained to do science well. So, this I know. Wait, what is that? Oh, yeah, this I do know. The hardest thing in life is falling in love and staying in love. But the important thing here is the second hardest thing in life is coming up with a truly original idea. This is the hardest thing in science. It's probably the hardest thing in life, but it's so satisfying when it happens. And the concept of this is not a mistake, this is nature trying to tell us something, and then thinking of a scientific approach to prove that is incredibly satisfying. So back to Phil. He didn't fire me. Instead, he said, let's get out of here. He got a job offer from University of Pennsylvania, and he said he wanted me to go with him. And I was like, I don't know about that. And so a friend of mine recommended that I talk to this gentleman, Ray Jesslin, who had come to the University of Utah from the Cold Spring Harbor Labs. And he agreed to take my appointment, and I was sitting with him, and he's like, well, what's on your mind? I'm like, well, I've been offered this job, and you know, my family's here, and I want to go to grad school here, and I'm talking about all this. And he said, stop. You're asking completely the wrong question. This is the basis of it. But the point was, if you really mean what you say in terms of being this scientist, in terms of understanding the world, then you have to go. There's no way you can stay. The perspective that you will gain from getting out of your comfort zone, with getting away from the things that you think that you know, will solidify your ability to study and to learn, and you'll develop new perspectives that will be impossible to develop if you stay here. He was right. It was one of the most important conversations in my life, and I told him so. So I went to Philly with Phil, and of course there he said to get back to work. I said, okay, it was very successful work. As we were mapping these genes, a little light bulb went on for me, and I realized, wait a minute, one of these days, every one of these genes is going to be mapped, and I'm going to be out of a job. What is it that I want to do next? And it was pretty obvious that the next thing to do was to understand how genes functioned. So I wanted to go to grad school. I went here, and I worked with this fellow, Oregon Health Sciences University. This is my advisor, doctoral advisor, Grover Bagby, the founding director of the Oregon Cancer Institute. He was the chief of Hem-Onc at the med school as well as at the Veterans Hospital. And he posed his question in a little bit of a different way. He said, let's explore. What do you want to do? I said, oh, by the way, I have this pet project. It's easy to articulate. I basically want to understand how leukemia happens. And that was it. I'm like, oh, that's a small question. But I said, yeah, let's study this. And so that got me interested in the blood. Ugh, blood. Blood is a fascinating, fascinating thing. For centuries, people have studied blood as a medical approach. A lot of bloodletting. I don't know if this is a barber or a surgeon. They were competing for control of medicine in those days. She looks pretty happy to be exsanguinated. This guy over here doesn't look quite as happy. Have thou a pox? Yes, take 2 of these. Ooh. Blood also has very interesting cultural significance. No matter what room I'm in, somebody always knows who this is. Vlad the Impaler. Vlad Tepes. The person. Sorry? The inspiration for Dracula. Blood can even mean romance. Nosferatu. That looked like me after my qualifying exam. Ooh, what's going on over here? What do you think? I'll give you a hint. This is a photo taken in sub-Saharan Africa, late 90s.
Malaria?
Not malaria. HIV. This person is in the last throes of AIDS, actually. The person died just a few hours after this photo was taken. In my lifetime, I went from seeing if you saw a drop of blood on the floor of the subway as just something ooh gross that you would step over to it being the most dangerous substance in the world. One of the great faculty members at Harvard, David Skadden, that I got to know and work with for quite a while, his main practice used to be HIV. And he said he would walk into the ward knowing that next week none of those people would be there. And that's the way it was. It was a treadmill of death. Blood is a fascinating tissue. I've studied the developmental biology of blood. This is like the United Colors of Benetton network of human and mouse hepatopoiesis. It goes through this sort of incredible journey, starting out in the yolk sac and the extra embryonic tissues and the placenta, and going through the liver and eventually into the bone marrow. It's really amazing how all of this is so tightly regulated. If we look at the blood as a tissue, it's pretty fascinating stuff. A skilled hematopathologist can recognize about 200 different blood cell types between mature cells, their progenitors, and cell types that are present only in certain diseases like buffalo cells or Burr cells or Howell Jolly bodies or the sea blue histiocyte. I have a friend here in the audience who knows about histiocytes. Why is your blood red? Well, for every 1,000 red blood cells, there's 1 white blood cell. And of the white blood cells, most of them are a cell called a neutrophil that's filled with granules. It tosses them out like little hand grenades to get rid of bacteria and other things, and they have a very short lifespan. And then there's all kinds of other cells. About half your blood is serum. It's the liquid component. As you're sitting here, you have a little over a gallon of it sloshing around. This older model, of hematopoiesis, where there's a blood stem cell that gives rise to all of these other blood cell types, is giving way, based on new research, to actually there being some heterogeneity up here. Some types of blood stem cells that make more of one kind than another, or have more of a predilection to making one type of blood cell than another, and they have a little bit of crosstalk, and there's this incredible dance of blood formation that I've heard referred to as hematopoetry, because it's so intricately choreographed and tightly genetically regulated. Have about 3 × 10 to the 13th cells, blood cells in your body, and it's an amazingly regenerative tissue. Millions per second. So as you're sitting here, 1 million cells, 2 million cells, 3 million cells, 4 million cells. That's how active your blood system is. These red blood cells, which fix oxygen, last about 120 days. Some of these granulocytes, the ones that throw out their little hand grenades, only last a few hours. Obviously, in your lymphoid system, you have cells that last for decades and decades. That's why your body remembers that you had measles as a child or chicken pox. Fascinating, fascinating stuff. It's also given way to some really terrific therapies, including hematopoietic stem cell transplantation. This gentleman, Don Thomas, I was lucky to get to meet him once. Got the Nobel Prize in 1990 for, in the 1950s, having this crazy idea that maybe you could transplant blood-forming activity from one person to another. It didn't work that well, but it worked well enough that science builds upon itself, and by the 80s, this type of transplantation was really starting to save people's lives. It's pretty simple. The person who has cells gives them to the person that doesn't. There's actually a picture from hematopoietic stem cell transplantation. This is Stanley. Stanley lived in Australia, had a particularly aggressive form of acute lymphocytic leukemia, which is the type of leukemia that most kids get, has a cure rate of like 98 or 99%. But for the 1% that aren't cured, it tends to be a very aggressive disease. Hematopoietic stem cell transplantation can often be the only hope. Stanley received that transplant from his donor. Here it just goes right into your arm. It's actually pretty anticlimactic. People are like, that's it? So sometimes they bang a drum or they clang cymbals to make it more exciting. Stanley relapsed and went through a second round of transplantation, and Stanley didn't make it. So despite the lives that are saved, biomedical research is about failures. It's about looking who hasn't been saved. It's about looking to what isn't known so that it can become a success. So I think the patron saint of biomedical researchers should be St. Jude, the patron saint of lost causes. In grad school, I studied these rare genetic diseases. This is one I studied for a long time, Fanconi anemia. It's a very rare disease. Has anybody here ever heard of Fanconi anemia? You have? Have you heard about it in an academic sense, or do you have experience with it? I'm sorry to hear that. How are they doing?
I don't know them very well.
Yeah. Most of the people I meet who have heard of Fanconi anemia have a story. And it's usually not a very good one. These kids tend to die of bone marrow failure. They just stop making blood before they'd be old enough to get a driver's license. Those that don't develop bone marrow failure are 30,000 times more likely to get leukemia than kids their same age. There's something about the genetic defect that leads to this condition that really makes the blood not function well, and that's what I studied in grad school. I know that there are folks here who aspire to go to medical school, so we have to do a little pathology to make it worth your while. You ready, Travis? So this is a normal bone marrow biopsy. Has anybody ever had one of these done? It's like, you know, when you take a core sample from a tree. I've had it done a couple of times. It's not my favorite thing, but it's not the worst thing. So you take that plug out and you can look at it. This is called a spicule. This is bone. All of these little empty spaces, this is fat. This is a very metabolically active tissue, and it has a high energy source rapidly available in close proximity to help engender that that biology in its fat. When a kid develops Fanconi anemia, this is what their marrow looks like. It's just got nothing in it. And if they flip over to leukemia, it's just so chock full of cells that there's no room for fat. In fact, if you look at these cells, they're very monotonous. They all look the same. If we look at a little bit of peripheral blood, this is, remember that 1,000-to-1 ratio, red blood cells to white blood cells. This is a granulocyte, that's a neutrophil. In leukemia, your white count goes up. And if you look at those white blood cells, they all look the same. They're part of that family tree, as my wife calls it, of hematopoiesis, but they're stuck. They can't mature. They divide. They live a long time. They crowd out all of the healthy cells, but they don't go on to be what they should be. So that's what I studied. And I'll be danged if I didn't get my PhD studying that. These are my brothers. Marty, Emily's pop. Dorkter is more like it, which is something he would have said to me. And my brother John, who thought that was a good one. I went on to a postdoctoral fellowship at the Whitehead Institute at MIT. Basically that same thing that I know about being able to live with failure but not regret. If you see the place you want to be, you need to go for it. I don't know why George accepted me into his lab, but he did. And it was a completely different experience. He didn't tell me what he wanted me to work on. He didn't assign me to a project. He gave me the key to the lab and said, do something amazing and let me know what you need. And it was this sandbox. And what an incredible scientific experience. The whole lab moved over to Children's Hospital and the Harvard Medical School. I moved with it. I stayed with George for quite a long time. I used to joke that I did neither well enough nor poorly enough to leave Harvard, so I'm still there these years later. But what I studied with George are stem cells. Let me tell you just a little bit about those. When I talk to students, I tell them that the most important word in stem cell biology is the F word. And I invite them to tell me what it is. And once in a while, somebody tells me a different F word that I didn't have in mind. No, it's function. Stem cells are defined functionally. You know it's a stem cell if it does two things, and those two things are this. When a stem cell divides, those two daughter cells are still stem cells. But one of them goes on to get a job. It has to mature. We call that differentiation. It goes on to become one of those other cell types that we talked about. And there are lots of different types of potential. Totipotent, this is like the fertilized egg. It makes every cell in the body as division goes forward, as well as the placenta. There are pluripotent cells that make all of the cells in the embryo, but not the part of the placenta. Multipotent cells, this would be like a blood stem cell. It makes a subset of cells, but not all. There are even unipotent stem cells that make one type of cell very robustly. Spermatogonial stem cells, a perfect example here. Nullipotency doesn't really describe a stem cell activity, but this is interesting. Some cancers are like this. They want to mature. They want to differentiate. But they can't do it. If you give them a little whiff of butyric acid, then they will differentiate, or retinoic acid. So somehow they're able to overcome that differentiation blockade, and some folks try to understand why that is to make better therapies. Let me see about skipping ahead here real quick. I knew I was going to talk too long. And come out over here. Oh great, I broke it. There. So the types of diseases that I was able to study with George are some of these. And what we tried to do was develop ways to study these very, very rare diseases of the blood in the laboratory, a quantitative way, even down to the level of biochemistry. And this is kind of an intractable problem. If you have a child with Fanconi anemia that walks into the clinic and you want to study their blood stem cells and try to understand why they're dying, they don't have any. That's why they have Fanconi anemia. And the ones they do have, they don't want to give them to you to study in the lab. They need them to make red blood cells. So how can you study these kind of conditions? Well, you can model them using stem cells. We did that with several. We did it with Fanconi anemia. Did it with a rare disease, dyskeratosis congenita, which is a defect of how the telomeres are organized, those little bits on the end of the chromosome, like that plastic thing on the end of a shoelace that keeps it from unraveling. Pretty much everybody knows about Down syndrome, but fewer people know that there's a specific type of leukemia that folks with Down syndrome can get. It's a very, very narrow type of blood disease that affects the cells that would go on to make platelets. We studied that. Even studied this really fascinating condition where kids will develop neutropenia. Those cells with the hand grenades will die, but they also lose the exocrine part of their pancreas, the part that produces digestive enzymes. And trying to understand how that happened was a really fun project. Most of these I worked on with a terrific guy, Asmund Topole, who's now a pediatric hem-onc fellow at University of California in San Francisco, the best person I've ever worked with in the lab. We approach these diseases using cellular reprogramming, which I'm sure folks here have heard about, but it's all about one question. If you have two different cells, if you have a heart cell and a neuron, for example, and they're in the same person, and you started as a single cell, then how do those cells become different from one another if they have the same DNA? Well, they flip those genetic switches. They use subsets of their DNA. It gets back to epigenetics. It's a really interesting experiment done by this fellow, Hal Weintraub, who was a terrific basketball player, I hear, at the Fred Hutch Cancer Research Center in Seattle. He was very interested in understanding these master regulatory cascades, these transcription factors that controlled muscle cell identity. And he was able to identify a gene, MyoD, because it was the fourth one he studied, A, B, C, and then D. And when that gene was overexpressed in fibroblasts, a cell from the skin, it essentially became a muscle cell. This was the first example of being able to reprogram or change the genetic trajectory, or the epigenetic trajectory, I should say, of a cell in development. Other people use this to great effect. I think most notably here, Shinya Yamanaka at Kyoto University, who in this incredible paper published in 2006, first described at the annual meeting of the International Stem Cell Society in Toronto, when he presented this, when he got up and said, I have a cocktail of genes, 4 genes, that are able to turn a mouse skin cell into a cell that's essentially the same as an embryonic stem cell, one that's able to make any cell in the body. And everybody thought, wow, that's fascinating. There's no way that's possible. But it was true. And it was true for the same reason that Hal Weintraub's experiment was successful. What Shinya was able to do with his colleague Kazutoshi Takahashi was to identify a master transcriptional regulator, a gene that when it's turned on activates dozens and dozens of other genes that are important for a specific cellular identity. And the cellular identity that he studied was from the very early stages of the embryo. He won the Lasker Award for this work, and he actually got the call to Stockholm for this, only 6 years after his basic discovery. In our lab, we quickly were able to use this technology to set up these genetic models of these rare diseases, only some of which we studied in our lab, but the rest we sent to other investigators around the world, because this is one of the most exciting parts about science as well. It's collaborative. And so the model is like this. You can take a tissue biopsy, pretty much any tissue with few exceptions, and using this cocktail that Yamanaka discovered, as well as some simpler and modified versions, you can turn these cells into ones that will grow indefinitely in the lab and are able to develop or mature into any other cell type in the body. So you basically take the cells you have and you can use them to make the cells you want. And if you're a guy like me who's interested in understanding where the first blood cell comes from in the body, it comes from some cell that's not a blood cell, but there's some genetic program that makes that happen. Now you can study this. You can study it in human biology. And you can study it in these very rare diseases. I mean, I can wave my arms around all day to make this point more emphatic. This is a revolution in our ability to study early human development. Maybe you can even make cells and put them back into patients. I wrote a really fun paper about this with my colleague Christine Mummery from Leiden University. And there's a little story behind this paper as well. I'm sure there are folks in the room who know what it's like when you promise to write a review. And then the deadline comes and you haven't written the review. And they give you an extension, and then that deadline passes and you haven't written the review. And I was actually down to the date where I was told in 1 week the paper has to be published, and if you don't have it, we're going to kill you. Well, the world sort of intervened. I woke up one morning and I heard on the radio that I was not allowed to leave the house because there had been a bombing at the Boston Marathon. And a few days later, the entire city, including Cambridge, where I live, was in lockdown. It was a shelter-in-place order while they looked for those two fellows. And while I sat there in my house, I wrote this paper. And so, this is what I know. Problems are almost always opportunities in disguise. I was once told by a colleague, never squander a great tragedy, because something important should come from it. That's how we learn. I've held lots and lots of other positions. I have this gene that when the word volunteer is said, my arm goes up. I don't know why, but it does. But I think the most important one is this one here at the very bottom, the shortest one. I love working with students. I love hearing what they want to do. I tell my students that I think the only thing that I continue to do of any use professionally is to introduce people to other people. And that's where fascinating stuff happens. And this is how I try to repay Wes Innes's parents for the gift that they gave to me. So this I know. From all of these different things that I've experienced and studied and tried to study, I have 3 rules for dealing with a difficult situation. I'm going to share them with you. I'm not even going to charge you. I'm going to get them for free. Rule number 1, identify your allies. I've shown several as I've gone through these comments up to this point. It's important to know who's on your side, where you can get the good advice. And usually when you go through that exercise, you also identify the people who might not be so much on your side. That's important information to have as well. Rule number 2, don't make things harder than they have to be. But importantly, don't make them easier than they have to be either. This is about knowing where your foundation is, where your line in the sand is. Okay, I will go to there, but I won't go any further because then I'm removing value. I'm removing what's important to me. You have to know who you are. The last one is the hardest one. And it's always be prepared to give up something you value in order to achieve something more important. You have to leave something on the table. You have to pay for what you get. You have to let the other person walk away with something. And I think how this last one in particular has been important in my professional life. From the time I was a little kid, I wanted to be a professor, but I'm not one. I'm not one because of a series of really interesting opportunities. But in the positions that I have now, and I have just a few more slides to tell you about that, I'm able to extend my reach. I don't work on my science anymore, but I work on lots of people's science. I work with students, I work with alumni, I work with fellows, and that's a very satisfying life. So my view of science has not been limited to the bench. What does it look like? It kind of looks like this. You know this story? The blind men and the elephant? They're all led into a room, and they've never experienced an elephant before, but they say, here's an elephant, tell us what it is. So this one grabs the ear and says, oh, an elephant is like the leaf of a large tree. And the person on the tusk says, oh, no, I don't know what you're smoking, but an elephant is like a sharp spear. The person out here on the trunk says, no, you guys are wrong. An elephant is like a serpent. The one on the leg says, no, an elephant is like the trunk of a tree. The one up here on the back says, no, you're wrong. An elephant is like a mighty boulder. And then the most unlucky person is this guy over here. An elephant is like a piece of rope. And the important concept here, of course, is that they're all right, but they're all wrong, because together they have a cumulative experience that actually says what the elephant is. And when I think of what science is, it's many things. It's these people. I've had about 650 students over the years from 140 different colleges and universities, including a terrific guy, Cody Tramp, from this university, who came through the program that I ran for 13 years. Notice what shirt I'm wearing. I know where I'm from. It's working with other teachers. Talk about amplifying your reach. If you teach a teacher, you teach every student they'll ever have. It's about engaging people. It's about being an advocate, about standing up for science, about trying to better understand what do people need, what do they want, how to get there, how to encourage policymakers, how to raise funds. I've even given talks in bars. People tend to get pretty animated when you're giving a talk in a bar. All of these people are about science. Policymakers. This is Adam Waters. He's Braveheart on the Royal Mile leading up to the castle in Edinburgh. He raises money. You have to pay him to take your picture with him. And every dime that he raises goes to breast cancer research in the UK because his wife died of breast cancer. He's raised millions of pounds being Braveheart in his after hours and on the weekend. He's making science happen. This is my pal Brooke Ellison, the first quadriplegic to graduate from Harvard. I was fortunate to be on her dissertation committee where she got a degree in sociology. I get all of the sort of one-off theses to read. She studied the role of executive power in setting science policy in the United States using human embryonic stem cell research as her case. And she's a delightful person. Lobbyists and advocates, members of the cloth. This was a terrific guy that I got to know who was a member of President George W. Bush's bioethics committee. I disagree with a lot of things that Bill thinks about, but he's a terrific guy. And of these, I'll just focus on this person. This is where the rubber meets the road for me. I'm fascinated by science, but I want it to do something. I want the world to be better than it is, and I just happen to be very lucky to study something that aspires to do that and that I'm interested in at the same time. This I know. The surest way to improve the quality of your own life is to improve the quality of someone else's. So, the last bit. There I was, minding my own business, being an administrator when an amazing thing happened. And this is what happened. My pal, George, had to go and become the new dean of the Harvard Medical School. And he called me up and said, would you like to work together again? And I said, yes, I have only one condition, and that I report directly to you. And he said, done. And then I asked him why he wanted to work with me. And he said, because he knew I would tell him if I thought he was wrong. And that's the basis of a terrific friendship. He said, let's get the band back together. I said, heck yeah. We both started on the same day, January 3rd. And this I know, friends are the family you make for yourself. It's important to have friends in this life, especially ones that you respect and that respect you. So I'll just end with some comments about what is this thing, the Harvard Medical School. It's the #1 medical school in the United States. It's been the #1 medical school since U.S. News & World Report has been doing its rankings. It's a pretty amazing place. Oh, it's also all of these things. It's such a complicated place. Harvard actually doesn't own its hospitals like most medical schools do. So instead we have these affiliated institutions that we have to work with. Some of these institutions do not like each other. In fact, they compete with one another for patients and for markets. And so you can imagine how fun that meeting is, to sit and try to get people to agree. But there are some terrific institutions here, the Dana-Farber, Joslin Diabetes, man's greatest hospital. Oh, I mean Massachusetts General Hospital, sorry. It means different things to different people. There's Gordon Hall, that's my office right there. Other than reporting directly to the dean, something that really is emblematic of our relationship is our offices are right next to each other and they have a door that connects them, like in a motel, so we can each hide over there if we need to. But that's how close the relationship is. We have more than 11,000 faculty. That's a few. 723 medical students. Our acceptance rate is 3.3%. About 165 matriculate. The rest of them go other places. We give a lot of aid. We have some terrific PhD programs. I heard recently that 1 in 7 postdocs in the United States is affiliated with one of the Harvard institutions. That's a pretty astonishing number. We have 34 HHMI faculty, 151 current members of the National Academy of Medicine, and 69 current members of the National Academy of Sciences. A little bit more for people that like to look at money. More than $250 million of grants and contracts per year, just at Harvard Medical School. If we look out to the greater Harvard diaspora, including all of those affiliated hospitals, it's nearly $3 billion of sponsored research income per year. It's an enormous enterprise. We had operating revenue for FY16 of $656 million and expenses of 692. Doesn't take an accounting major to realize that there's a difference between those and it's not in the right direction, so we operate with a structural deficit. Our faculty and our programs are bigger than our return on endowment and our ability to raise funds. And so that's something that we work on. Quickly, what do I do as chief of staff? Gosh, it really depends on the day. I'd say it falls under one overarching principle and three functions. The big picture is I try to put myself in George's place, and I think, would I want somebody if it was me to take care of this? And if the answer is yes, then I take care of it. I reach out throughout that universe of affiliated faculty, of core faculty, this incredible cadre of deans and associate deans and assistant deans and vice assistant deans, second class, and make things happen. It's a terrific, terrific group of people. The three functions were taught to me by Mitch Weiss, who was Mayor Tom Menino's chief of staff for years, one of the longest serving mayors in the U.S., the great mayor of Boston. Number one is ideas. You have to think up things that you need to do, and then you have to think about who can help you do them. Results in accountability. If somebody promises the dean something, somebody has to go out and check on it, and that's me. Help break log jams if they're there. Figure out how come it hasn't happened. Also, one of my favorite things to give praise and recognition when things do happen. People work hard. The last one is the feeding and the watering of the dean, sometimes quite literally. I'm going to give you some insights into some current projects and problems so you know what we're dealing with. I'm sure I don't need to tell folks in the room that the federal landscape is changing almost daily. Some of these are very central to the way a medical school and its affiliated hospitals work. Three here, the uncertainty of future government funding. Are we going to be able to have grants the way that we have? Possible hurdles for indirect cost recovery, which I'm sure some folks in this room know quite a bit about, but it's basically the overhead that allows you to operate the university in which the research happens. And there's talk about cutting that to a fraction of its current level. This would cripple universities all across the nation. New regulations for certain types of research, the immigration policy, including the recent decision about DACA, hits Harvard Medical School. I have friends who are not able to come back into the country after the immigration ban. And we have students who are facing a really uncertain future who are dreamers in this country, who've only ever known this country and now wonder what their future is going to be like, even though they study at a place like Harvard. Not trying to get into politics. I'm just talking about the problems that we have to deal with at the university. They're very real. Managing this incredible array of people is complicated. How do we stimulate research in a deficit situation? You have to find ways to make that happen. You can't cut your way out of a deficit. You have to grow your way out of a deficit, which is a bit of a paradox. Leading people who probably don't want to be led. Those hospital CEOs who would really like to see that other hospital just burst into flames and burn to the ground, but somehow trying to get them to agree to common ground. Increasing diversity across the board. Harvard has made some great strides. We don't have nearly enough women on the senior faculty and in senior positions of leadership, as well as people from underrepresented minorities and physicians that are underrepresented in medicine. We take this very seriously. And then the big one, looking beyond today. You spend your whole day answering email, and all of it's important. But you have to find time to carve out to think, what should we be doing? What do we want the school to look like in a year, in 5 years, in 50 years? And then make sure that you put some of those wheels in motion. So this I know. The best job you'll ever have is the one you make for yourself. I hope you're making it. I just want to thank some people. Thank you, Travis, for inviting me. I was really pleased to get your email. It's just a delight to be here. I'm glad I get to meet you in person. The folks in the dean's office, Patrick, you've been a terrific pal. I look forward to seeing what we're going to do to fill in that hole with some bricks and mortar. Wonderful people in the College of Science office. I've mentioned several people who helped me along the way. I'm really pleased that some of them are in this room. Others aren't. Beth Hood is a terrific person. Jim Bowman I mentioned. John Simmons, who was chair when I was an undergraduate here, was a terrific guy. Fran Tishner. Wonderful people at this university. And in every talk I've ever given, I thank the true experts, the patients and their families. They literally give of themselves so that other people can have a better life. And I thank you for your time.
So I have a question to start, actually.
What do you got?
So how do you, not, I don't know, get intimidated? I guess I imagine Harvard to be such a cutthroat environment, and maybe it's not. Maybe I'm wrong there, but
Some places are.
how do you
honest.
not just get kind of intimidated by the caliber of people that you're working with all the time? And I don't know. Does that make sense?
It does make sense. So there's this thing called imposter syndrome. There's so many of the students I advise, especially in the summer program. I mean, they come in, about half the students were from Harvard, but the other half, they were from St. Olaf College in Minnesota, or Bunker Hill Community College, or Scottsdale Community College in Arizona, or these little colleges that the only time I ever heard of that college was when somebody walked up and said, "Hi, I'm from Hamilton College." And I'm like, "Oh, where's that?" And then it's really fun, and they're all like, "What are we supposed to do here?" It's like, you were invited. Do work. Who do you want to meet? What do you want to study? Don't be intimidated. There's no reason to be intimidated. I was intimidated when I came here. I mean, this is a terrific place. I mean, think of your colleagues that you know. Everybody starts somewhere. And the people that make those interactions about them, those are not the people to work with. And there are, those labs. I could go to the board and I could write down some of those labs at Harvard, at MIT, at the different places I've been. But some people like those environments. That's not an environment that works well for me. I like one where people are collaborative, where they hold their science to a high bar. Well, they will attack your science in lab meeting because they want it to be better. So go to that place and study what you want to study and forget about the rest of it.
Okay, thank you.
Sure. What's on your mind?
So now that the Human Genome Project is over, what do you think is the next big project?
So now that the Human Genome Project is over, what's the next big project? Well, we still don't know how it works. That whole area of epigenetics, how this cell versus that cell decides to use these genes and not those genes, and how all of that is orchestrated and regulated, is incredibly complicated. In fact, I think it's the type of problem that until recently, you could sit and think about it and stare at the sun or look at the lake and come up with interesting ideas about how you think the world works, but it's really only because of the parallel fields of biology and computation that I think that we're able to truly analyze the types of data sets that are required to understand epigenetic regulation. And so that remains the cutting edge. And what's fascinating about that is one of my favorite biologists of many, Hans Spemann, who got the Nobel Prize in, I forget what year, but he studied cellular organization. There's this thing called the Spemann organizer that's about how different cells come together. And the types of questions that he asked in his lab in 1925 are the same types of questions that people ask today. Good questions sort of happen in their own time. And they're usually able to be capitalized upon because of timing. And it's either the right person comes along and thinks about it in a different way. And one of my favorite quotes is, "The greatest obstacle to discovery is the illusion of knowledge." That person who comes into a field and either they don't know what it's about because they're brand new, because they're a freshman and they're wondering, or they come in from a different field and they don't have that foundation of biases that other people have learned, and so they ask questions that everyone should have asked. Maybe they ask it in a different way. The flip side of that is an enabling technology, a tool that allows you to do something that you could think about before, but you couldn't reduce it to practice. And there are lots of great examples. I think one of the foremost is microscopy, and then lots and lots of other permutations from there. So all the hot labs that I know of in genetics, there's still a lot to be understood about the genome. We could take a poll right now among the scientists sitting over here on this side about what is a gene. Does it follow the central dogma, DNA, RNA, protein, or microRNA's genes? or long non-coding RNase genes. They don't make proteins, but they can produce a phenotype. I mean, there's a lot of fascinating genetics, but there's no loss, no lack of interesting questions. What kind of questions are you interested in?
I heard of the brain initiative, so I thought that was like the next big thing, but I agree with you, there's a lot of research to be done there.
Yeah, I have a bias for genetics because that's what I studied, but the brain initiative is fascinating. I mean, where are your memories? Where's your consciousness? What is it? What are your emotions? How are your experiences stored? Are they real? I mean, the brain is the most complicated machine that I'm aware of, and I even have a smartwatch on. So, fascinating stuff. Really incredible neuroscience and systems biology. I think one of the marks of a person who's a dyed-in-the-wool scientist is no matter what question somebody starts talking about, they'll be interested in it. And they'll come up with a question. You know, we could start to talk about petal variegation in tulips. And I know these guys will be interested in something about that, as will I. Yes, sir.
You touched a little bit on the diversity that Harvard really prides itself on, which is fantastic.
Mm-hmm.
But how would you suggest diversifying yourself as a white middle-class male?
Yeah. So how do I consider my own diversity? This is interesting, and it's an important question. So a couple of things I would say. First, there's a little story. When I was in graduate school, I made this mistake of being out of town when they held the graduate student elections. And I was elected president because I was I think the only person up there. So let that be a lesson to you. Always be in town unless you want the job. But as part of that job, I used to give an annual talk to underrepresented minority students who were interested in graduate school and medical school. So you can imagine me standing up there talking to these kids about science. And there was this time this kid said, he raised his hand, I'm like, what's your question? And he said, why are you here? And I said, well, I grew up in Utah, but I'm not a Mormon. And he said, okay, you're fine. So there is this aspect of diversity depends. But even though I come from, hardworking people, but sort of difficult, I still had opportunities that I don't think would have been mine if I looked different than I do, if I was a different gender than I am. I have to face those realities. And you can't let it keep you back, but you can't squander it at the same time. And so how do I deal with diversity? I try to create diversity. I try not to take for granted how I was born. I can't change that, but I can make the most of what I've been given and try to open the door for other people. Maybe to tell you another little story here. I'm sorry, I'm so full of stories. We recently hired an executive dean for the medical school. This is the number 2 person and the lead administrative person for the medical school. And there's this whole phalanx of people, like 20 people on this committee, that we make all of these interviewees run this gauntlet. And we ask them all the same questions so you can actually compare apples to apples for their responses. It's actually the way people should be interviewed. The only thing that would make it more fair is if you couldn't see them and couldn't hear them so you could truly have a blind appraisal of the person's abilities. But be that as it may, one of the questions that was always asked was about diversity. And man, what an incredible array of answers. Some people who would just say, yes, diversity is important. Next question. I'm being facetious, but something close to that, and other people that would give more examples. And I would sit there and think, what do I do about this? And so I work on projects, like even things as maybe different as looking at the artwork around the medical school and saying, wow, there's a lot of old white guys in these pictures. Meeting with people, trying to come up with resources, looking through our vast collections to maybe take down that 3rd portrait of Oliver Wendell Holmes and to put up one of Harriet Hardy because she was awesome. Harold Amos, African-American professor at Harvard Medical School. So things like that. But I think the most important thing is you have to go out and find people. You have to walk it like you talk it. And I've worked very hard, not as hard as I can, not as hard as I'm going to. to actually find those students who are here and they want to be here, and then you help them. You do it one at a time, you really mean it, and you stick with them. So I don't know if that's a good answer to your question, but that's the answer I have. Thank you. Yes, sir.
You talked about how in your stem cell research, you take a skin biopsy and eventually produce stem cells for that person. And then you mentioned briefly putting them back into the subject you took the stem cells from. How close is that science of, say, growing a heart for someone that has heart failure, or growing pancreas, and putting that back into someone that organ is failing in the body?
Yeah, so the question is, how close is this cellular reprogramming science to actually having clinical impact? In some areas, it's very close. And so you ask, I'll focus on two. You ask about making a heart and being useful for treatment of diabetes. The first one is farther away. If you think about making an entire heart, a heart is actually a complicated organ. It's actually like 2 or 3 organs that grow together. It has elements of neural crest, it has elements of mesoderm. It's actually an incredible structure with a fantastically intricate developmental biology. So making an actual heart is very complicated, although there are people that are working on that. And there are people that are working on just patches of cardiomyocyte tissue to put on areas that have been infarcted or that have died in heart attack. So people are working very actively on that. It's not my specific area, but I hear people talk about within the next few years having clinical trials in this area. I try not to hang timelines on things because usually in a room there's somebody who's a lot more interested in that timeline than other people for a very personal reason. But it's exciting work and there's a lot going on. Diabetes is closer. Why is diabetes closer than the heart? Because a single cell type dies in diabetes. I think it's probably an autoimmune attack against the insulin-producing cells of the pancreas, the beta cells and the islets of Langerhans. So if you think about the need to engineer a complicated structure with many interdigitating cell types or replacing one type of cell, the bar is lower for the one type of cell, right? And for example, there's a guy at Harvard, Doug Melton. He was the faculty chair of the department I directed for some years. He is very close. In fact, he's close enough that he's formed a company to be able to take this platform through clinical trials. This is one of the, I think, more interesting areas of biomedical research, and one that can be very difficult to talk about depending upon the room that you're in, but no hospital ever made a pill, right? They don't manufacture stuff. And so those partnerships between the research university and the private sector is required actually to be able to take new treatments through to the clinic. And the fact that he's formed a company around this, and I'm not a shareholder, I don't have any part of the company, so I have no conflict here other than I think it's fascinating, and I think that it means that it's very close. And he's published some papers in the last few years to show that using this cellular reprogramming technology in a diabetic mouse model, that he can use mouse cells or human cells, and that those cells are glucose responsive, insulin secreting, and normalize the blood sugar levels of mice. And now he's partnering with people at MIT, working on biomaterials to develop a matrix with a highly modified alginate, basically, from seaweed, so that it doesn't stimulate an immune response. It contains the cells, and they can be implanted like in the peritoneal space, or they're working on devices that can be implanted under the skin. It doesn't have to be in the pancreas. It just has to have a blood supply, and it's able to regulate blood sugar. And so that's a lot closer. Keep your eyes on that space. It's fascinating. I hope that was a decent answer. Thank you for your question. Yes, sir.
So I believe a lot of us here are pre-meds, and I'm just wondering your opinion on where you see the medical field going in the future, maybe where you see it, maybe where you would hope that would go. What do you see in maybe our future as we go on to medical school and above?
Go for it. The importance of primary care is only going to become more important. I think that some of the more interesting terrain that has to be conquered is around affordability and insurability. But if you look on the medical side, I mean, there are great careers out there for people who want to hang out their shingle in their community. Important careers, especially if you want to go to an underserved area. You know, that's something I would encourage you to do. And this is the worst kind of advice. You should do something that I never considered for myself, right? But it's important stuff. But things like electronic medical records, the type of interface between virtual reality and surgery, for example, like, you know, about robotic surgery and distant surgery, but ways to approach the surgical subject in a way that's, you know, more like VR. I mean, I hear of all types of really fascinating things. And around the medical school, I actually don't know how many departments there are. There are 10 basic science departments, including healthcare, policy, and global health and social medicine. But then you get out into the hospitals. I mean, there are dozens and dozens of departments, and there are multiple departments of neurology, for example, where people are doing research as well as providing primary care. I think the biggest hurdle that I hear about, again, to get back to what I said a minute ago from sitting in the dean's office, is what is the future of affordable health care going to look like? And what's that going to mean for people receiving care? And what's it going to mean for those delivering care? How much of your time are you going to spend filling out forms as opposed to providing care? And how, even at the medical school, we can continue to nurture that fascination and that devotion to service that people come into the field with, because physician burnout is also a very big problem. I think especially for people who practice in remote areas where you're not just a pediatrician, you're an everything, right? You're the insert in this space, ologist. Sorry, I don't have better advice than that. What type of medicine are you interested in?
I'm interested in general practice, actually.
Yeah?
You're a family doctor or a pediatrician.
It's important work. You'll touch many lives. Let's have snacks. Okay, thank you for your time.